Well, welcome everyone to our monthly AMS seminar. I'm Jonathan Plett. I am the president of the society, and I have had a bit of a break as I've been traveling quite a bit recently. So thank you very much to my committee members who have kept this going in my absence. So this month we have the pleasure of having Ryan O'Donnell, who's going to be talking about their PhD research. Ryan has an interesting background. I loved reading um, the biography sent through that Ryan started as an operatic singer and found science more exciting. I hope he talks more about that because that's fascinating. I would think being up in the limelight would be much more entertaining and singing your heart out. But now Ryan is in the limelight for the work that they've been doing down at ANU, working with Celeste Lind and others in Orchid mycorrhiza. So with that, I will pass it over to Ryan. Thank you very much.
Thank you very much for that introduction. So g'day everyone. Thanks the AMS for having me. Thanks Camille for the invitation to present to you all today. I feel like I'm a bit of a newcomer to the Australasian mycological arena, so I'm really grateful to the Society for making me feel very welcome and giving me this opportunity to put my work out there. So welcome to my talk, Discordance Down Under, combining phylogenomics and fungal symbioses to detangle difficult nodes in a diverse tribe of Australian terrestrial orchids. This is a summary of some of my PhD work, which I presented at this year's International Mycological Congress in Maastricht in August, and it's currently in review, so watch this space. Before I begin, I'd just like to give an acknowledgement of country, acknowledge the traditional owners of the land on which we meet today, which for me is the Ngunnawal and Ngambri peoples, and I'd like to pay my respects to elders past, present, and emerging. So I think I'll start by answering a question that you're probably all thinking, which is, who am I? So I'm Ryan. I'm a final year PhD student working under the supervision of Celeste Lind at the Australian National University here in Canberra. I came to biology through plants, and my master's research focused on the systematics and taxonomy of Australian mint bushes. So I ostensibly started my PhD as a botanist. I was looking for PhD projects and stumbled upon Celeste's lab on the ANU website, and I saw that her group was looking at orchid fungal co-evolution. And this was a bit left of field for me at the time, considering I was mainly interested in plant systematics, but I thought it sounded interesting enough, and I reached out to Celeste, and 3 years later, here we are. So while I started as a botanist, over time, I think I've become more and more interested in the fungal side of things. So I'm sort of pivoting more towards mycology these days. Now, my PhD work is focused on the systematics and taxonomy of the Ceratobasidiaceae, specifically the genus Rhizoctonia, and its various synonyms like Ceratobasidium, Thanatophorus, and Ceratorhiza. But the reason that I've honed in on the Ceratobasidiaceae in particular is because they're pivotal players in orchid mycorrhizal relationships, which is what I've spent a large part of my PhD researching. So, the evolution of plants and fungi is inextricably linked. Around 90% of plants form mycorrhizal relationships, and 75% of plants partner with arbuscular mycorrhizal fungi, or AM fungi. Orchids, on the other hand, do things a bit differently. So apologies, mycologists, I will have to talk a little bit about plants for a while here. Bear with me. So the Orchidaceae, or orchids, represent the second largest plant family on Earth, second only to the daisies. With approximately 28,000 species found on every continent except Antarctica, the orchids encompass around 8% of all flowering plants on Earth. In Australia, we've got about 1,700 species, depending on your taxonomic flavor. But the thing I find so interesting about these orchids is that they are obligate symbionts. So over time, orchid seeds have evolved to become smaller and smaller and smaller to the point that they're now basically dust-like and microscopic. So there is an obvious pro to this adaptation, which is that the seeds can be dispersed very easily and widely just on the wind. But the trade-off is that there is no more endosperm left within the seed. So the workarounds that orchids have stumbled upon is the recruitment of ubiquitous saprotrophic fungi as symbionts. Rather than the usual AM fungi, putatively orchid mycorrhizal families encompass several ectomycorrhizal fungal families here in green, a few other saprotrophic or ectomycorrhizal families here in grey, but the majority of orchid mycorrhizal relationships are formed with cryptic basidiomycetes, broadly referred to as rhizoctonias, in the families Ceratobasidiaceae, Tulissinolaceae, and Serendipitaceae. Without these fungi, these orchids are unable to even germinate from seed. So these fungi will colonize the orchid seeds, provide them with nutrients they can access from the soil, and one day the fungus might receive something in return from the orchid. And it's hypothesized that this recruitment of these fungi as symbionts opened up a whole new suite of nutritional niches and microhabitats to the orchids. And it's hypothesized that this relationship is just one of the factors driving their staggering diversity today. These relationships range from being very broad, gregarious, to being highly specific. And nowhere is this specificity more evident than the Australian terrestrial orchid flora. So the Australian terrestrial orchids are dominated by the tribe Diurideae, which contains about 900 to 1,000 species, represented by most of the genera on this slide, and the subtribe Terrestrialidae from the sister tribe Chronichidae, shown in the bottom right there. So today I'll be talking mainly about the Diurideae. This tribe contains some of the world's most charismatic orchids, including Rhizoanthella, the rare parasitic mycoheterotrophic orchid, which is known only from Australia, which spends its entire life underground, and an unusually high number of species that engage in what's called sexually deceptive pollination. So sexually deceptive pollination is notably a rewardless pollination system where flowers have evolved to look and smell like female insects in order to dupe male insects into attempting to mate with the flower and inadvertently pollinating them in the process. Now, this phenomenon has been documented in about 150 species across at least 9 genera, but there's almost certainly more that are undocumented. In many of these cases, each orchid species has a one-to-one single species of pollinator that it attracts. So in Australia, we not only have a high rate of above-ground biotic niche specificity, but we also seem to have a high rate of below-ground specificity too. Now, we've known about this propensity towards specificity in Australian terrestrial orchids for some time now, since this extensive isolation and germination work by Walkup in the 1960s and 70s. Walkup observed that several species and entire genera of orchid would often partner with only 1 to 4 fungal OTUs, usually from a single fungal family. And on this basis, he posited that there was mycorrhizal specificity ranging from the species to possibly subtribe scale. Australian orchids, the terrestrial orchids, appear to partner with an average of only 2 OTUs per species, which is extremely narrow compared to what's been observed in European systems, which partner with an average of about 13 OTUs per species. But why such specificity has evolved both above and below ground in Australia is still unknown. Now, Walkup observed that closely related orchids appear to share similar fungal partners, and he hypothesized that these relationships might be phylogenetically structured and may reflect the orchid phylogeny. But our ability to interrogate these questions has been hampered somewhat by a lack of phylogenetic resolution in the Diurideae phylogeny. So since the first molecular phylogeny of the tribe, published by Kores et al. In 2001, the placement of several subtribes has been proved particularly challenging, namely the Acianthinae and the Prazophilinae. As you can see in this figure here, with adapted trees of all major phylogenetic reconstructions of the tribe to date, the placement of these 2 subtribes remains pretty variable regardless of how many loci you use, what kind of DNA, like nuclear or chloroplast, or method of analysis used. So why is this phylogeny so difficult to resolve? Long and short of it is that we don't know. But that's something that I've been quite interested in teasing apart. Previous authors have suggested several reasons why the tribe has been challenging, including rapid speciation and subsequent incomplete lineage sorting, introgression or hybridization, extinction, or the presence of these parasitic mycoheterotrophic lineages like Rhizoanthella. So, to come back to Rhizoanthella, I mentioned that it is a mycoheterotrophic parasite which lives out the entirety of its lifespan underground. So Rhizoanthella is in a tripartite relationship with a couple of OTUs of Rhizoctonia and some species of Melaleuca, and maybe other things. But as Rhizoanthella appears to get all of its nutrition from the fungus it's partnered with, it has lost the ability to photosynthesize, hence its white achlorophyllous appearance. Consequently, the genome of its chloroplast has become highly reduced and degraded, which has produced great difficulty when attempting to figure out the phylogenetic affinities of this thing. While there's been a whole plastid genome for Rhizoanthella sequenced, until now, we've not had genome scale nuclear data available. So, on one hand, we can say, well, we can't investigate these evolutionary questions until we have a fully resolved phylogeny for the Diurideae. But it seems like regardless how much genetic information we throw at this problem, we're not going to gain a satisfactory answer. So I thought, can we flip this problem and work backwards? Can we use these fungal relationships to support certain phylogenetic hypotheses in spite of these complex evolutionary histories? So, for this study, we aim to infer and date a robust phylogeny for the Diurideae and investigate potential causes of phylogenetic discordance, determine the dominant fungal partners for genera within the tribe, reconstruct how and when fungal symbiont preferences and specificity evolved, and finally, determine whether these fungal symbiont preferences do indeed reflect the orchid phylogeny. So in the interest of time, I'm going to gloss over the methods a bit, but I'll provide a brief summary. So we use sequences generated for the Diurideae, generated using a custom bait set along with some transcriptomes, and we combine these with publicly available genomes, transcriptomes, and target capture sequences for the Orchidaceae and monocots more broadly in order to provide outgroups for fossil calibration points. Using these data, we used HivePiper for reference-based assembly and paralog resolution or orthology inference using the Paragon pipeline. We then used IQ-tree to infer gene trees and Astral to produce a coalescent-aware summary tree. And then we fossil calibrated this topology and used MCMC tree to do Bayesian divergence time estimation. Once we had a tree, we had to then identify the dominant fungal partners for each of our target genera. So to do this, we synthesized decades worth of published fungal sequence data, germination, and morphological data. So we mined fungal ITS sequences from GenBank and other published studies, which have been isolated and uploaded for all of our target genera, reblasted them to make sure everything was what it said it was. It wasn't. But we reassigned everything to the family level. We then coded the dominant fungal partners as characters and performed marginal ancestral state reconstruction with phytools across several different phylogenetic hypotheses, which we observed in our data. So, time for some results. So here is our calibrated time tree with 13 fossil calibration points, including 6 orchid fossils. It makes it the most densely calibrated Orchidaceae tree to date. We can see that the split between the predominantly Australian Diurideae and the predominantly South American Chronichidae was inferred to have occurred in the Eocene around 52 million years ago, with the Diurideae crown age inferred at about 46 million years ago, which is consistent with the breakup of Australia, Antarctica, and South America. All subtribes were inferred to have stem ages between 46 to 36 million years ago, with all subtribes having split within this period of about 10 million years, all having diverged by the Eocene-Oligocene boundary. Now, the branches here are colored by gene concordance factor, which is the proportion of gene trees that agree with the species tree, with purple showing high support and yellow showing low support. But you'll see that echoing previous phylogenomic studies of this group, we found high levels of phylogenetic discordance, particularly along the backbone of the tribe here. So I know that there are some of you who are not phylogeneticists, so I will give a brief rundown on some of the concepts that I'll be discussing, namely phylogenetic discordance and its causes. So what is phylogenetic discordance? Each gene tells a different evolutionary story. Ideally, each of these genes tells a similar evolutionary story, but more often than not, this isn't the case. So put simply, phylogenetic discordance is when gene trees don't necessarily agree with our hypothetical species tree. There are several evolutionary demographic processes that cause phylogenetic discordance, and these include introgression or hybridization, incomplete lineage sorting, which I'll discuss in detail on the next couple of slides, or paralogy or gene duplication and loss. So I'm going to focus on incomplete lineage sorting for a bit. But incomplete lineage sorting, or ILS, also known as deep coalescence or the retention of ancestral polymorphisms, occurs when loci that are polymorphic at the time of species divergence randomly fix afterwards in a pattern that doesn't necessarily reflect the evolutionary history of that lineage. So while ILS is a temporary process, it leads to permanent genomic signatures, and it's often associated with rapid radiations. So how do we distinguish between ILS and other demographic processes in our data? So a simple way is by comparing alternate topology frequencies and determining the ratio of sites or genes which support the main topology versus alternate topologies. So since under the assumptions of ILS, we can expect ILS to act randomly across all members of a clade, we can expect a roughly equal frequency of each alternate topology. Other demographic processes, namely introgression, for instance, will produce uneven alternate topology frequencies, as these processes generally are characterized by gene flow between two lineages. So, what do the alternate topology frequencies at our difficult nodes tell us in our data? Our results suggest that there is no significant difference between alternate topology frequencies at these nodes, meaning that they're roughly equal. And as they're roughly equal under the assumptions I outlined just then, we can say that ILS is probably the most likely driver of phylogenetic discordance in the Diurideae at these difficult nodes. So this signature of ILS, combined with the fact that all of these subtribes within the Diurideae had diverged within a 10 million year period, are all results consistent with the presence of a rapid radiation in the tribe's evolutionary history. So moving on to the fungal character mapping, our synthesis of over 70 years worth of published research into these groups, which encompasses detailed germination studies and over 3,000 fungal ITS sequences, revealed an unprecedented level of fungal niche specialization. So we can see here that entire genera, subtribes, and even groups of closely related subtribes partner almost exclusively with a single fungal family. Additionally, transitions to these near exclusive relationships were inferred to have occurred in a small window of approximately about 5 million years between 46 to 41 million years ago. So I only showed one topology in the previous slide for the sake of simplicity, but as I said, we've repeated our ancestral state reconstructions for each dominant alternate topology observed in our data. But regardless of which topology we used, we saw the same general pattern, which was that the ancestral diurid was most likely more gregarious with respect to its fungal associations, but over time, members of the group have become increasingly specialized in their associations. So I'm going to come back to this figure for a little bit to talk about something that I think is a bit interesting. So looking at the backbone of the Diurideae here, we can see that these nodes, which were the most challenging to resolve in our phylogenies, also appeared to coincide with the major fungal symbiont shifts in the tribe. So looking at this, it almost begs the question, did fungal niche specialization facilitate a rapid diversification in the Diurideae? So my hypothesis is that fungal niche differentiation early in the tribe's evolutionary history may represent a key innovation where specializing on different fungal groups opened up a wider range of nutritional niches, which subsequently facilitated rapid diversification, resulting in the patterns of ILS that we see here. We know from experimental studies on several Australian orchids that orchid mycorrhizal fungi are capable of accessing different environmental nutrients with varying ability and preference. And several Australian plant groups have developed specialized root adaptations to cope with the continent's ancient nutrient-poor soils. So it's possible that the evolutionary trend towards increasingly specialized symbiont associations in the Australian terrestrial orchid flora is another example of an Australian plant group adapting to depauperate soils. So following this, I'm hoping to continue this work by testing whether these fungal symbiont shifts are indeed associated with diversification rate shifts within the Diurideae. So that's the next part of the project. But to come back to our original question that I posed at the beginning, can fungal associations be used to support the phylogenetic placement of problematic groups? Let's see. So first, Rhizoanthella. As I mentioned, for the first time, we sequenced a transcriptome for Rhizoanthella, which we were able to include in our analyses. And also for the first time, our results have recovered Rhizoanthella as sister to Prasophyllum and Genoplysium. So these genera all associate exclusively with the family Diversification. So we have molecular and mycorrhizal data which support placement of these Diversification specialists together as sister genera. Next, the Diurideae. So this subtribe has previously been recovered as sister to the subtribe Caledoninii, which associates exclusively with the Serendipitaceae. Diurideae, however, appears to associate exclusively with Tulissinolaceae. So its placement here alongside the other Tulissinolaceae specialists is parsimonious. And finally, Acanthaene. While some members of the Acanthaene do seem to partner with members of the Tulissinolaceae, we can see that a number of these genera are capable of partnering with Serendipitaceae. But that ability to partner with Serendipitaceae is completely absent in the Tulissinolaceae specialist clade. So the fact that it's able to partner with Serendipitaceae means that its placement as sister to the Serendipitaceae specialist in Caledoninii is sound. So to summarize everything, we synthesized decades worth of research into the mycorrhizal relationships of the Australian terrestrial orchid flora, and we found an unprecedented level of fungal niche specialization at a much deeper scale than previously recognized. So these associations were likely broader in the past, but have become increasingly specialized over time. After almost 70 years, we've provided support for Walkup's hypothesis that orchid symbiont preferences do indeed reflect the orchid host phylogeny. And we can use these relationships to provide support for the placement of phylogenetically difficult groups. And with that, thank you. I'd just like to acknowledge a few people here who have contributed to this project in various ways, and also to the plant and fungal research community. This work wouldn't have been possible if people didn't upload their data to GenBank and other public databases. So it really represents a synthesis of so many people's work over the years, and I don't think it would have been possible if we didn't have those sort of resources. But yeah, got some contact details here if you feel like reaching out, saying good day. Yeah, thanks for having me.
Brilliant. Thank you so much, Ryan. That was great. Does the audience have any questions? You can pop it into the Q&A box and we can see that and Ryan can answer. While people are doing that, a question that I had, and I apologize if I miss this. So as you know, you go from a very generalist ancestor or hypothetical ancestor and then come into this more specialization. Do you have any ideas or have you been looking at what's happening within the genome of these?
Do you mean with respect to the orchids or the fungi?
The fungi.
Looking at like how the fungal genomes um, have
Yeah,
changed.
so yeah, so if it's getting more specialized, is it losing genes? Is it gaining genes? Because you said you did transcriptomic work. So I'm just curious
So
to know. Yep.
yeah, the transcriptome stuff was for the orchid side of things.
Okay, sorry.
Um, yeah, but I mean, one of my chapters will be looking at the sort of genomics of some of the rhizoctonias that associate with some of the Australian orchids. So that's something that I am hoping to look at, and I know Celeste is doing some other work with the genomes of the other things like some of the Tulacinella and Serendipitous. So
Mm.
um, it's sort of an open space. There hasn't been a huge amount of work that's been done on the genomics of the orchid mycorrhizal fungi. There are in other orchid groups, there have been like some genomic or transcriptomic studies sort of looking at that interface. And there are sort of several putative mechanisms that have been suggested for what's happening at that interface, but it's still a wide open question, really.
Yeah.
But I think that the extreme specificity that we can see here in the Australian systems, I think lends itself really nicely to answering some of these questions, and since there are these really defined niche shifts. Um, I think it presents a real nice model system to look at those questions.
No, absolutely. A question from Tom May. Hi, Ryan. Lovely work. Do you have plans to carry out this kind of analysis at species by species level for the orchids in relation to the set of individual species of fungi that each plant species is associated with, and doing similar analyses of the phylogeny?
Um, there have been several smaller studies over time which have done that. I know particularly with the Drakini and yeah, some of the Caledonia as well. Um, it's a bit of a trickier question to do it like at the species scale with each species of um orchid, because like there are quite a number that don't have fungal data available, and there are so many species. I mean, like, it's taken. I think our data set represents there's data for around 400 orchid species in there, which is a fraction of what the Australian orchid flora is. So, and that's taken like, over 70 years to accrue that kind of information. So I guess to get that sort of same level of coverage for every species would be, take us another however many years. Um, but yeah, like I do want to look at it at the sort of like which OTUs are associating with each genus, at least from a sort of preliminary glance. It does appear that different genera even appear to partner with different OTUs to other genera. So like even there, like there is a bit of that fungal niche partitioning, if you want to call it that. But yeah, it would be. I'd love to, keep working on it, and yeah, doing that at that scale. And actually looking at what OTUs are sort of there, and sort of binning them in that respect. And how it sort of relates to things like biomes and biome shifts and things like that. Um, too many things to do and not enough PhD time to do it.
Well, you have your whole career laid out for you now then. Um, John Durnally asks, he says, sorry, I missed the start of your talk, Ryan, and you may have addressed this. Why do some orchids only have a single OMF symbiont? And is it related to physiological efficiency?
Um, Short answer is I don't think we know. Um, yeah, it's sort of unclear what them at the moment, like what exactly is governing that specificity. Like I know the genus Kyloglottis, for instance, yeah, only has a single Tulisnella that it partners with. So it's sort of unknown why that is the case. Um, yeah, I think until we sort of understand like the mechanism of what's actually driving things like the host symbiont recognition and things like that, we probably won't know. I mean, there have been several suggestions as to like what's happening, um, whether it's like um yeah, some molecular mechanism governing that host recognition, or whether the fungus is producing some sort of effector to bypass the plant immune um, recognition and getting in that way. And um, the fungi sort of live in the orchids as those pelotons, and it's sort of unclear, like, whether like the orchids are acquiring the nutrition like through the fungi itself, or because they're lysing the fungal pelotons, and that's sort of how they're gaining that nutrition. So it might have something to do with the ability of the plants to like engage with those. Yeah, I don't know. It's a big question, and lots and lots of work to be done.
Question following up on one of the things you had talked about before, and you were mentioning that, you went through and pulled all of this data and found that a number of them, based on phylogeny now and what we know, were misannotated as to what, or misidentified as to what they were. Do you have plans now to now go back into like the base database or other databases to begin seeing, where else you might find these fungi?
Um, what do you mean? Sorry.
Because I'm just curious. So I'm thinking to like data that my group is playing around with when we're looking at like pine roots, eucalyptus roots,
Yeah.
and we're finding, oidodendron, we're finding serendipity, we're finding other things that technically should just be orchid mycorrhiza. So I'm curious if you're now that you have done a better phylogeny of these orchid-associated fungi, of going back now into other data sets that are publicly available to see, what other plants they might associate with. Are they as specific as we might have assumed or not?
Yeah, I mean, a lot of them are sort of fairly ubiquitous things that occur in soil and detritus and whatever. I know that the Tulisnellas are difficult because they don't, in a lot of those soil DNA things, they don't amplify terribly well. They're very divergent, and they do lots of weird stuff. So it's difficult to sort of look at that question, I guess, with that group in one. The rhizoctonias and the serendipitas maybe. I know that so when I was reblasting everything, I did find that like the blast like RefSeq database and even Unite, it did have difficulty identifying things that were definitely serendipita, for instance. Like the type sequences of serendipita, which we know are definitely serendipita. And it had difficulty identifying them as serendipita. So, yeah, I sort of had to create a custom version of the RefSeq database, which had all the types for the serendipitas and the sebacenas and the rhizoctonias and tulisnellas that weren't in there, so that it could more confidently assign that. So um, I don't know. I think I'd probably just express caution with like interpreting GenBank blast results a lot of the time, because um, a lot of it's mislabeled, and as I said, some of these groups are a bit difficult to sort of assign to that level. So um, I think it's a area which needs a bit of investigation. Yeah, for sure.
Great. So John has another one. Any perspectives on rare OMF impacting on the rarity of certain native orchid species?
So there is some work looking at this. I believe some of Ryan Phillips' work and some of Nushka Ryder's stuff. Um, Basically, they seem to suggest that like orchid rarity is not really associated with not really bound by the fungus. So can you just read the question again? Sorry. Um,
Any perspectives on rare OMF impacting the rarity of
Oh.
certain native orchid species?
yeah. A lot of the time, those rare orchids sort of like tend to partner with OTUs that are fairly common. And so yeah, it doesn't seem to be a limiting factor, but I guess it's also difficult to sort of determine how rare some of these fungal OTUs are, because yeah, we don't really know what their distribution is. We kind of have an idea, through some of that, the broader like soil work, but um, yeah, it's kind of hard. I would like to like take look at some of that data and see, um, if it can provide a better picture on like the distribution um, of some of these OTUs and whether there are some that are like genuinely rare. I mean, there do seem to be some which only it can only be found in like certain like tiny little bits of substrate, like sphagnum moss or something. So I mean that I'm sure there's heaps of stuff that's hiding out there that we don't know, but it doesn't seem to be a constraint for the orchid rarity based on the work that's out there.
Cool. Tom just has a comment. He has a comment on the serendipita that crop up under in other samples. Sorry. He had a look at Unite recently, and just for serendipita, there are a staggering over 600 species level species hypotheses at 3%, almost all of which are unnamed. So there are a lot of undescribed fungi within these groups. So more stuff to dig up.
Yes. Look, the serendipity is absolutely like some people in our group actually are working on like on the serendipitas, but um like Eriacalis, for instance, they isolated like 30 different OTUs from Eriacalis alone, and all of them are undescribed, which is crazy. Like, um, yeah. So there within Australia, there does seem to be a lot of serendipitas that need names. A lot
Yeah,
of
for sure.
all of them really, like um yeah, a lot of the rhizoctonias and like Mark Freestone did look at the sort of rhizoctonia diversity across Australia, um the orchid rhizoctonias, as in Rhizoctonia sensu stricto. Um and like, there's at least 26 things that need names, probably more. um yeah, which will probably like be even more once you like use more than just ITS. Yeah, the serendipitas, there's heaps, at least 30, probably more. Tulisnella, who knows? Because oh, there's heaps of Tulisnellas actually.
Great. Well, thank you very much. Thank you very much for your time today, Ryan. That was really great. Um, Look forward to seeing this in print. And thank you, everyone who showed up today, and for those who will be watching this later on YouTube. Remember, we'll be back again next month at the end of November, but we also will be at ESA. So we look forward to seeing those who are going to be able to make it to Melbourne. Remember, we will also have a screening of a movie there. So please do come out and enjoy us and say good day to us as well. With that, I will say bye to everyone, and thanks again, Ryan.
Thanks so much for having me.
Bye for now.